right? So talked about sugars, their structures, what they can be used for as energy and as structural elements. And lipids are very much the same, right? Lipids, as you see in chapter 22, can be used for energy through their oxidation, but lipids also serve a structural role. So what is a lipid? What are they made of? And what kinds of structure do they make up? So they make up biological membranes. Biological membranes are simply just structures that separate two different compartments. For example, a plasma membrane separates the inside of the cell from the outside of the cell. What's important to note is that both the compartments that are separated by these biological membranes are typically aqueous. Right? So we have an aqueous environment inside the cell, mostly water, separated from the outside of the cell, which is also mostly water. And this is important because the cell needs to maintain necessarily an in equilibrium with the environment. Right? What happens inside the cell must necessarily be different than what's going on outside the cell, and the only way to accomplish this is by separating out the two components. And biological membranes have a few common features, right? They're closed boundaries between compartments, obviously. They consist of mostly lipids, which we'll talk about today, and proteins. Proteins are embedded within these membranes in a few different ways that we'll talk about today. Lipids have hydrophilic and hydrophobic moieties and form bilayers, right? So what do we mean by moiety? A moiety is simply just a part of a greater thing. In this case, we can break down a lipid into two moieties, a hydrophilic and a hydrophobic, or a polar and non-polar moiety. Many other compounds have moieties as parts. Proteins have specific functions within the membranes, right? So these proteins that are embedded in the membranes, they have functions, and these are what carry out most of the processes that membranes carry out. Right? So membranes aren't inert. They have to either pump molecules into or out of the cell to establish concentration gradients. They have to transduce signals from outside of the cell to inside of the cell, and so on. All of these processes are carried out by the proteins that are embedded in the membrane. Membranes associated with non-covalent forces, and notably, they self-assemble, right? So these membranes come together and they form spontaneously because of the most stable configuration of that system, as we'll see in just a minute. So there is nothing necessary in order to form a membrane aside from just having lipids in an aqueous environment. Membranes are asymmetric, so one side of this membrane will look necessarily different than the other side of the membrane. This is what helps to distinguish between in and out, or one side and the other, right? So if you look at the bottom side of a membrane versus the top side of the membrane, they'll have different populations of proteins, different orientations of proteins, in order to move things in the correct direction or to transduce signals in the correct direction. And membranes are fluid, right? They aren't, a solid sphere surrounding a cell, for example. In the instance of the plasma membrane, they're kind of wavy, right? They have a fluidity to them, and there's an optimal fluidity that a membrane must adopt. If it's too solid, right, too much like a solid sphere, well, that affects how these embedded membrane proteins can carry out their functions. Okay, so we need them to be able to diffuse laterally, as we'll see, but if they're too fluid, well, then the membrane just falls apart. So how does the membrane adopt its most optimal fluidity, right? Kind of that Goldilocks zone where it's not too solid and not too fluid. So first, what is a lipid made up of? Well, a lipid is made up of a polar component, which we'll get to in a minute, but the non-polar component in this case are always going to be these fatty acids, right? So what is a fatty acid? Fatty acids are essentially just oxidized hydrocarbons. And we can think of it as just a hydrocarbon that terminates in a carboxyl group, right? So here we have the simplest, or at least the most common fatty acid, palmitate. We see that it's just made up of 16 carbons, and it terminates in a carboxyl. That's it, right? And so these fatty acids are amphipathic. They have a non-polar component in the form of the hydrocarbon tail, and they have a polar component in the form of the carboxyl at the end. And just like amino acids, right? Remember, that's the pKa for the carboxyl group of an amino acid? 2, 3, somewhere around there. The same is true here, right? That's the pKa generally for a carboxyl group. So at pH 7.4, which is where we're going to find most of these fatty acids, they will exist in their ionized state. So we refer to them as their ionized name, palmitate instead of palmitic acid, or in this case, oleate instead of oleic acid. In this case, this is just another fatty acid, though here we have what we call an unsaturation, a double bond. A monounsaturated fatty acid has one double bond. A diunsaturated fatty acid has two, triunsaturated, so on and so on. Okay? But why do we say it's unsaturated? Because this is not as saturated as it could be with electrons and protons. In other words, hydrogens. Okay? Here in palmitate, this is a fully saturated fatty acid. It's got as many hydrogens, in other words, protons and electrons, as it can have. Right? Each one of these is a methylene group terminated with a methyl on the other end. Here we're missing 2 electrons and 2 protons. Right? We have 2 methyl groups here. So this we call an unsaturation. And no matter what the identity is here of the hydrocarbon tail, right, it always terminates in carboxyl, so we always have a polar and non-polar component. We can name these systematically, right? It's very easy to name these systematically. In this case, palmitate has 16 carbons, okay? So we would name it as 16 colon number of double bonds in the case of palmitate 0. So palmitate is the 16 colon fatty acid, 16 carbon, 0 double bonds. So you can tell right off the bat that it's fully saturated. In the case of oleate, it's a little bit more complicated, right? So here we would have 18 carbons colon 1. We have 1 double bond. Now, where is that double bond? Well, that's denoted by the symbol delta, okay? 18 colon 1, delta 9, right here, there's a formatting mistake. The number should be in a superscript, and the number is describing on which carbon, starting from the carboxyl, the double bond first appears. So 1, 2, 3, 4, 5, 6, 7, 8, 9. The double bond appears on carbon 9. So this is an 18 colon 1 delta 9 fatty acid. We can tell that it's monounsaturated, and we know that the unsaturation occurs on carbon 9. Now, we'll get to the naming, the more systematic naming in just a second, but unfortunately this is the first, or actually the second after the carbohydrates, the second time that we're seeing that these fatty acids have common names, right? They were named for either the color they have or a reaction they were found in, or most commonly the source that they were first isolated from. So palmitate was isolated from palm oil, oleate was isolated from olive oil, and these are just historical common names that unfortunately there's no way around just memorizing. Okay, so these common names you'll see whether you become a doctor, nurse, or researcher, right? These are just things that you have to know, but there is a more systematic way to name them. All right. Okay, so fatty acid nomenclature. We always start numbering at the carboxyl. Oh, here it is. We always start numbering at the carboxyl carbon. This carbon number 1, 2, 3, so on and so on. Carbons number 2 and carbon number 3 are always referred to as the alpha and beta carbons. Okay, and this is going to become especially useful once we get to chapter 22 and discuss the beta oxidation of fatty acids. This is how the cell extracts energy from these fatty acids. The reason it's called beta oxidation is because it's the beta carbon that's going to be oxidized. Right? So it's the beta oxidation of the fatty acid. We can also start numbering at the methyl terminus, right? So the other end. In this case, we refer to the methyl terminus as the omega carbon. And in this case, it doesn't really matter what number of carbons there are. Always the terminal methyl group is referred to as the omega carbon. So in palmitate, carbon number 16 is the omega carbon, whereas in oleate, carbon number 18 is the omega carbon. And the reason we do this is because sometimes the double bond or the unsaturation occurs at a certain number of carbons away, not from the carboxyl, but specifically from the methyl. Okay, famously we have the omega-3 fatty acids. Omega-3 fatty acids are an example of something called an essential fatty acid. They're necessary for proper brain development. Okay, but our bodies are incapable of synthesizing omega-3 fatty acids, so it's essential that we consume them in our diet. There is no other way for us to get them. Omega-3 fatty acids were first identified because children deficient in their intake of omega-3 fatty acids tend to display developmental delays. Okay, it hinders their brain development, right? So sometimes it's useful to refer to the unsaturation from the omega carbon. Now the systematic name for a fatty acid is a lot easier to kind of understand than the common name. The common name, again, is just straight rote memorization, which I always hate. But at least the systematic name has rules. It's named for the hydrocarbon on which the fatty acid is based, right? So for example, palmitate has 16 carbons, so that would correspond to hexadecane, right? So palmitate has a systematic name, hexadec. Oh, where are we at? Palmitate, hexadecanoate. So we just replaced the last E in hexadecane with O8, right? Hexadecanoate. And in this case, because it's in the ionized form, it's hexadecanoate instead of hexadecanoic acid, right? So that's very simple. When we have an unsaturation, though, okay, we have to be careful. We have to first specify in what configuration the double bond is, cis or trans. The delta, just like we've seen before, where we describe where the double bond is. But in this case, instead of anoate, right, we just say hexadecanoate, enoate instead of anoate, right? So that A is replaced with an E before the decade in this case, right? So it's a small change, but it is important. So in this case, we have for oleate, right, we have oxidecane for 18 carbons. We replace the last E with O8, so oxidecanoate, but we have an unsaturation, so the A in decane is replaced with an E, oxidecanoate, right, cis delta 9. We go back here, of course, we have a cis double bond at carbon 9, it's oxidecanoate, and there we go, right? So the names here are very simple to follow. We can have two double bonds, so we can have a diunsaturated fatty acid. In this case, here again, linoleate, two double bonds, 18 carbons, octadecadienoate, right? Or trienoate and so on, right? This is just the rules of naming the systematic fatty acid. If we have more than two double bonds, instead of saying cis, 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 or cis, or trans, 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 right? We can just write all cis. Delta 9, delta 12, delta 15, octadecatrienoate, right? So very simple. We can also name them, of course, going from the omega carbon. Palmitate is an omega-7 fatty acid, and so on and so on. Okay? All right, now unsaturated fatty acids cannot be conjugated, right? What does this mean? That means that there always needs to be a methylene group, at least one methylene group in between the double bonds, right? So we cannot have a conjugated fatty acid. Now, there is nothing against the rules chemically of having a conjugated fatty acid, okay? It's just the way that they're synthesized by biological systems that we don't see conjugated fatty acids. Fatty acids will always have at least one methylene group in between the double bonds. So if we said here, right, linolenate, which is what we're looking at here, which is a delta 9, 12, and 15, right? 9, 12, and 15. Notice there's one methylene group in between all these. An invalid fatty acid would be to say if we had a delta 9, 11, 15. In that case, we have a double bond here, and a double bond here, and a double bond here. In that case, we would have a conjugated system between these two double bonds, which is not a valid fatty acid structure. So we always need to have a methylene between, okay? Now, in reality, what we see is that the double bonds that we encounter are almost always in the cis configuration, okay? A trans configuration leads to problems, let's say, right? So a trans fatty acid, I don't know if you guys are old enough to remember, you know, the trans fats scare of the mid-2000s, right? Where everybody was banning trans fats and all this. The reason for this is that trans fats, well, we'll get to it in just a minute, but trans fats lead to other undesirable consequences. Even though there's nothing structurally really wrong with them, they resemble saturated fatty acids, but they have other consequences that are less desirable. So a lipid then is made up of mostly fatty acids, okay, but we also need now our polar component, because once we incorporate our fatty acids into our lipids, that carboxyl is now not as strong a polar group, right? And this also changes the structure of a lipid compared to a fatty acid, which makes them uniquely suited to form biological membranes, as we'll see in just a second here. And membrane lipids we can break down into three different categories: phospholipids, glycolipids, and cholesterol, right? Oh, and sphingolipids, which we'll talk about, but it's not in this list. So 4 categories: phospholipids, which are the most common, glycolipids, sphingolipids, and cholesterol. So a phospholipid is made up of 2 fatty acids on a glycerol scaffold. Okay, so glycerol is simply 3 carbons. Each carbon has a hydroxyl, right? So COH, COH, COH. the first two carbons, carbons number 1 and 2, are linked to their fatty acids via an ester bond. They're esterified to a fatty acid, like we see here, right? So here R1 is just whatever fatty acid this is. It could be palmitate, it could be oleate, linolenate, doesn't matter, right? But they will always be linked via the carboxyl carbon to carbon number 1 and carbon number 2 of glycerol, which is what we're seeing here, right? Via an ester bond. Carbon number 3 of the glycerol will always be connected to a phosphoryl group. Okay, so we have two fatty acids on carbons number 1 and 2, and a phosphoryl group on carbon number 3. This is the simplest phospholipid, and it's called phosphatidate, or phosphatidic acid in its ionized form, or in its unionized form. But in its ionized form, which it is at pH 7.4, we call this phosphatidate. All phospholipids are based on this simplest phospholipid, phosphatidate, which is why, as we'll see, all phospholipids are named phosphatidyl X, phosphatidyl Y, right? Because they are derivatives of this phosphatidate. And if we examine the structure here of phosphatidate, notice what we have. Two fatty acid tails over here, which are non-polar. And on this side, we have a very polar phosphoryl group. So we have our two fatty acids, two non-polar tails with a very polar head group, in this case phosphate. But on this phosphate and all the derivatives of phosphatidic acid, we attach an alcohol. And this gives us greater variety in the types of phospholipids that we can create. For example, we have serine. This can be linked by a phosphoester bond, actually phosphodiester bond, to carbon number 3, glycerol. We can have ethanolamine, choline, glycerol, which is interesting because glycerol also forms the backbone here. So we'll see what that looks like in just a second. And inositol, which is particularly important because phosphorylated derivatives of inositol can also be used as the alcohol in a phospholipid. And as we'll see in chapter 13 or 14, these phosphorylated derivatives of inositol can serve as signaling molecules during signal transduction. Chapter 14, I believe. Okay? So what matters is that there is a hydroxyl or alcohol, right, that can be linked onto the phosphoryl group here. And so what we end up are with phosphatidic acid derivatives, phosphatidyl serine, right? Notice here we have serine, the carboxyl, C-alpha, and alpha amino group. Here, this red oxygen used to be the hydroxyl at the end of serine. Phosphatidylcholine, phosphatidyl-ethanolamine, phosphatidyl-inositol. Okay. Notably, what we have down here is diphosphatidyl-glycerol, also called cardiolipid. Right? Here we have carbon number 1 and 2 of glycerol is esterified to fatty acids 1 and 2. Carbon number 3 obviously connected to a phosphoryl group. In this case, the alcohol is glycerol. Right? Well, what do we know here? Well, the other carbon number 3 of glycerol in this case can also be linked to a phosphoryl group, and this phosphoryl group can be linked to carbon number 3 of a third glycerol molecule. And this third glycerol molecule, carbons number 2 and 1, are also esterified now to fatty acids 3 and 4. So what we have here is a structure that's got 4 fatty acids, okay, and they're all pointed away like this. So when we categorize cardiolipid, or diphosphatidylglycerol, when we categorize this as amphipathic, remember what amphipathic means? Got a polar group and a non-polar group on opposite ends. We think that cardiolipin is amphipathic, as a fatty acid is. Okay, so how would we configure this so that it is amphipathic?
Fatty acids are pointing this way, and the middle of the molecule towards the bottom. There's a negative charge there. Could be amphipathic.
Right, so that's exactly what happens. Cardiolipin, though it looks like we just have 4 fatty acids going out that way, right? This can still serve and does as an important membrane lipid if we take these, right, and rotate them up so that we have 4 fatty acids going up. Down here, what do we have? We have this OH and we have these phosphoryl groups, so we do still have a polar head group. So now we have kind of a different configuration than the normal phosphatidylipid or phospholipid that we talked about with only 2 fatty acids. This cardiolipid, right, where would we expect to find this? Look at the name. Cardiolipid.
In
heart, right? And what does the heart need a lot of? Constantly, famously. What is the heart always doing, ideally? Pumping blood. So what does it need constantly in order to do that? Energy, right? So if it needs a lot of energy, what does it need a lot of in terms of cellular structures or organelles? It needs a lot of ATP, and where is the ATP made? In the mitochondria, it's the powerhouse of the cell, right? So, where do we find a lot of this cardiolipin? Well, we find it in tissues that are enriched in mitochondria, okay? Or we find it in the heart, which is enriched in mitochondria, I should say. And what we see is that cardiolipin is in fact enriched in the inner mitochondrial membrane of mitochondria. And the reason for this is that cardiolipin helps to stabilize the complexes of the electron transport chain, which are also found in the inner mitochondrial membrane. And we'll talk about chapter 18. And cardiolipin helps to stabilize these, right? So wherever we have the electron transport chain, in other words, the inner mitochondrial membrane, we need cardiolipin in order to stabilize the tertiary and quaternary structures of those complexes. Now, a different type of phospholipid we can see is sphingosine. Sphingosine is not built on a glycerol, or sphingolipids rather, are not built on a glycerol backbone. Instead, they're built on a sphingosine backbone, right? So what do we have here? Sphingosine. Well, we have already something that kind of resembles a fatty acid, already part of the scaffold of the backbone. What we have is a amino group here and a hydroxyl here. So this is analogous, this right here is analogous to carbon number 3 of glycerol. And this amino group is analogous to carbon number 1 of glycerol. So instead of attaching a fatty acid via an ester bond to this amino group, it's not possible. Instead, we can attach it via an amide bond here, right? So here now we have the hydrocarbon tail of sphingomyelin. We have the hydrocarbon tail of one fatty acid. On carbon number 3, we can attach a phosphoryl group, and by a phosphodiester bond, we can now attach our second alcohol. Okay, so in this case, if this alcohol is choline, right, well, this becomes specifically sphingomyelin, right? Sphingomyelin is our sphingosine backbone with a fatty acid, a phosphoryl group, and specifically choline. We find sphingomyelin in the myelin sheaths surrounding neurons. They help to isolate, electrically isolate these neurons so that signal transduction can occur down the axon. Okay. Now, where do we have it? The blue and green, right? So the sphingosine plus just a fatty acid, we call that a ceramide, right? The reason is that just a fatty acid linked via an amide bond to sphingosine is a waxy substance, right? Does anybody speak Spanish here? Right, so what's the Spanish word for wax? It's a tough one. Yeah, cera. That's the Spanish word for wax, and it's an amide, so ceramide, right? Comes from the Latin, right? So speaking Spanish is very helpful if you want to pick up Latin words here and there. So a ceramide is a waxy amide, and in fact, yes, when we have this sphingosine esterified only, or linked by an amide bond, not esterified, to a fatty acid, this forms a waxy substance. Here we wouldn't have the phosphoryl group or the alcohol, this would just be an OH. And we find ceramides very often in the extracellular matrix. Okay, they help to, as you would imagine, right, if you have a waxy substance all in your face or elsewhere in your body, right, you can actually help to firm up that skin, right? So many cosmetic products, or not many, but some cosmetic products do include specifically ceramides because they do help, in theory, to firm up the skin, right? By providing this waxy ceramide substance. It's already part of the extracellular matrix, but you do tend to lose as you get older. Okay. Now, we also have glycolipids and cholesterol. Glycolipids are built on sphingolipids, right? So they very much resemble a sphingolipid. We have our sphingosine backbone and amide bond to a fatty acid, but instead of a phosphoryl group and alcohol, we have a sugar subunit, or a sugar moiety, I should say. Okay? So we can have either a monosaccharide or a polysaccharide. They all encompass these glycolipids. They have different functions. Here we see an example of a glycolipid called cerebroside. If the sugar component is specifically either a glucose monomer or galactose monomer, we call this a cerebroside. We find these in the cell membranes of many brain cells, as you would imagine, cerebroside. Cholesterol is kind of the odd man out here. It doesn't resemble really any of the membrane lipids that we've talked about, either glycolipids, sphingolipids, or phospholipids. They always have those two tails, whether it's part of sphingosine or a fatty acid, and they have a polar head group. Cholesterol looks nothing like that, right? We have this four-cyclic structure here, which is built on the steroid nucleus, right? And the steroid nucleus is actually very useful, right? So many hormones like testosterone and estrogen are built on the steroid nucleus. Cholesterol is built on the steroid nucleus. But specifically this carbon, okay, has a hydroxyl. This hydroxyl here, that's our polar head group, okay? So while it may not look like that, and it looks vastly different than all the other membrane lipids, right? We do have a polar head group, and now we have this hydrophobic part over here, right? This is cholesterol. Now cholesterol, in the context of a membrane, or the cell membrane specifically, doesn't function the same as either a glycolipid, sphingolipid, or phospholipid. Those three are what make up the vast majority of the membrane. It's what gives them their structure, as we'll see in just a minute here, a couple slides. Cholesterol is used to regulate the membrane fluidity of that membrane, okay, at least in eukaryotes. This is how we mainly regulate our membrane fluidity. It helps to kind of buffer changes in fluidity. It reduces the effect of temperature and other things on fluidity, so it helps to maintain that optimal fluidity of these membranes. Archaeolipids, we'll not really talk about too much. There's just a few differences. Notice they look very much the same, right? They're still built on glycerol, but they're inverted, so instead of carbons 1 and 2 holding the hydrocarbon tails, in this case we have carbons 2 and 3 holding on to the hydrocarbon tails, and they're linked via ether bonds, not ester bonds, right? So these ether bonds are important because in archaea, which are famously mostly extremophiles, these ether bonds help to stabilize, or they're not as susceptible to hydrolysis as the ester bonds, right? So in cases where they're found in extreme pH situations or in high heat situations, right? These membrane lipids, because they're linked via ether bonds, are more stable than they would be if they were esterified glycerol. And then over here in carbon number 1, same deal. We just have it flipped. Instead of on carbon number 3, we have the phosphoryl group on carbon number 1 linked to some alcohol, in this case, ethanol. Oh, and the hydrocarbon tails in archaeal phospholipids are also branched. Okay, so in the case for eukaryotic phospholipids, because they're built on unbranched fatty acids, the tails will also always be unbranched. They can be unsaturated, but they will always be linear. They will never be branched. Here we have almost uniformly branched nonpolar tails. Reason being that these little methyl groups here, these methyl branches, they provide extra contact surface, you could say, right? So they increase the van der Waals forces between the membrane lipids to hold those membranes together, especially useful, right, when we're living in, say, a hydrothermal vent. Now, membrane phospholipids, again, are antipathic, and we can depict them kind of like this, right? So we have our two nonpolar tails and our polar head group here. And we can see, right, this looks vaguely familiar. Here we have a phosphoglyceride, so here's a phosphoryl group, our two fatty acid tails. Here we have some alcohol, right? Let's call it ethanolamine, it looks like. Ethanolamine. So here we have a phosphoglyceride. Here we have sphingomyelin, all right? So here's the tail of sphingosine. Here's our fatty acid. Here's our phosphoryl group and our alcohol. The archaeolipid, notice, is just bigger because of those branches, right? But they all follow this same kind of pattern, right? We have our two tails and our polar head group over here, so we just depict them like this as shorthand. Now, if you look at, actually not this slide. But if you look at some literature, right, so for example, you can look up on Google, you know, how does the lipid nanoparticle that delivers the COVID-19 mRNA vaccine look like? Well, this mRNA is packaged inside of a vesicle, which we'll talk about in the next slide. And there they go into, you know, the different types of phospholipids that make up these lipid nanoparticles, right? In that case, they're always drawn like this, but elaborated on somehow over here, so you can tell apart the different types of phospholipids, right? But generally, right, if we don't care the identity of the specific phospholipids, we just draw them as a little ball with two little legs. All right. Now, how do these behave in water? Well, fatty acids and our phospholipids, right? So here we have what we call a micelle. A micelle forms when you suspend fatty acids in aqueous solution, right? And why is this? Well, we again remember fatty acids terminate in a carboxyl. They have that nonpolar tail, and so that nonpolar tail doesn't really want to interact with all that water, but the polar head group does. So if we have just this in solution, well, that's, you know, not very favorable. Remember, that hydrophobic effect, these water molecules are forming a cage around the nonpolar tail, but they're forming hydrogen bonds with the carboxyls. So all of these water molecules that are forming the cage around the nonpolar tail, they don't want to be there. So how can we free up those water molecules forming the cage over the nonpolar tail? Well, by forming a micelle. Okay, so all of the polar carboxyls start interacting with each other and with the water surrounding them, and they point their tails inward, so that the tails are now interacting with other tails and not with water, specifically not with water. So the water is excluded, and so it's free to form hydrogen bonds now with the carboxyl groups or with other water molecules. This is more entropically favorable than having them be just free in solution. So this is the most stable configuration of fatty acids in aqueous solution. And because it's the most stable, micelles form spontaneously. In other words, they self-assemble. There is nothing required to form a micelle, okay? And where do we find these free fatty acids that form micelles? Well, we find these in soap, okay? So this is simply just what soap is. It's a mixture of fatty acids or molecules that look like fatty acids. For example, lauryl sulfate or sodium lauryl sulfate. This is present in almost every shampoo and body wash. It's just soap, right? This is the most common detergent that you will use, right? If you look at the ingredients list for any shampoo, you'll see this lauryl sulfate. And this looks very much the same as a fatty acid. It's got 12 carbons, right? And it terminates in a sulfate group instead of carboxyl, but the same principle holds, right? That sulfate, which is negatively charged, is polar. We have 12 carbons saturated, so these don't want to interact with water, so they form micelles in solution. And these interior here of the micelle, well, now that can hold on to nonpolar molecules and suspend them in emulsion in aqueous solution, right? So this is how soaps work. You can make soaps also by hydrolyzing triacylglycerides, which is animal fat. You can make them by hydrolyzing membrane lipids, right? So you free up those fatty acids so that now you have fatty acids in solution. But this, if we look at the cross-section, looks like this, right, is not very big. The radius is limited by simply the length of the fatty acid, right? So these can't form very big structures at all yet.
So, like this type of structure, if it's like in soap, right, that destroys like bacteria cells?
Yes, it would destroy bacterial cells, right? So if you add SDS, this disrupts the plasma membrane, as we'll see in just a minute here, right? Reason being, again, because on the inside here, right, we can hold nonpolar material, right? So this negates the hydrophobic effect, which is what drives the formation of both micelles and these bilayer sheets, which we're about to talk about, right? So these micelles, again, their radius is limited by the length of the fatty acid. We can't form anything bigger, right? What would happen if we formed anything bigger? Well, now we would have more, let's say, more fatty acids. But we have all our tails pointed towards the inside, but what's on the inside? Well, it'd just be more water, so we're not any better off than what we started with, okay? Instead, what we see is that they form these micelles where there is no water on the inside, it's excluded. Now, lipids, phospholipids, sphingolipids, glycolipids, they can't form micelles because they have these two fatty acids. their cross-section, right, is more rectangular as opposed to these fatty acids, which is more triangular, right? So these triangles assemble into, circles, which are the micelles. But because these lipids are more rectangular, they assemble into what we see as sheets. Okay, now if this were the case, well now we have, right, so ignore these guys on the bottom, we have all of these non-polar tails just pointed down now, interacting with the water, and that's still not stable. So in order to exclude water from interacting with the bottoms of all these tails, well now, more tails from more membrane lipids will start to interact with these. So we exclude water entirely from the interior of this bilayer sheet. So that still leaves the edges, right? So if we imagine that this is a perfect square, right, made up of these membrane lipids, well, we'll have water interacting here, along the sides, and along the back. To exclude that water, right, the edges of this sheet form a tube, okay, but then we still have the ends of the tubes that are interacting with the water, so those close up, and what we form is something called a liposome, right? And a liposome is kind of the model for how the cell membrane looks. We have two layers, right? We have the phospholipid bilayer, okay? And this is now separating two aqueous compartments, right? We have the aqueous environment interacting with the polar head group, and we have the aqueous interior interacting with the polar head groups. Each half of this bilayer we call a leaflet, right? So the phospholipid bilayer is made up of two leaflets, right? Closed around each other in order to form a liposome. Now a liposome can be very small, okay, or it can be very big. In this case, we are not limited by the length of the fatty acid making up the lipid, right? We can just keep adding more and more lipids into our liposome in order to make it grow and grow and grow, and it will always form roughly a sphere, right? Unless there are other components inside that are forcing it to be shaped differently. For example, bacteria, which is not a sphere, instead they're usually kind of tubes. This is caused by the cytoskeleton on the inside. But the phospholipids alone will always form a sphere. The radius of the sphere just depends on how many phospholipids are in that liposome, or that liposome membrane. So we'll skip about soaps and detergents. It's just interesting chemistry, right? So this is how, I guess we won't skip it now. It's just interesting chemistry. So this is how soaps were initially made, right? We took animal fat, which are made up of largely triacylglycerides, right? So it's 3 fatty acids esterified to each carbon 1, 2, and 3 of a glycerol. This is how fats are stored in the body. Again, like we'll talk about in chapter 22, but animal fats are stored exactly as well. We have animal fat, right? So this triacylglyceride is susceptible, the ester bonds here are susceptible to hydrolysis when the pH is high and the temperature is high. So what they would do is that they would take these animal fats and just boil them in lye, which is sodium hydroxide, and these would hydrolyze these bonds here. So we'd end up with sodium salts, so fatty acids, and glycerol, right? So that's it. After you boil it in lye, you let it cool down. When it cools down, right, depending on the identity of these fatty acids, especially if they're fully saturated, right, they will cool down and solidify, and now what you have is a bar of soap, right? So you add to that some essential oils or some odorants, and bingo bango, you got yourselves an Etsy shop, right? And detergents have other uses. They can be used to soften water or other things like that. I won't get too much into that, but this is an interesting read on the slide if you're interested, which you very well might not be. Vesicles, okay? So these micelles that form in the context of biological membranes, we call these a vesicle. And a vesicle is simply just a very small liposome in a way, okay? So what are these vesicles and what are they used for? Well, vesicles are used biologically by the body. For example, when acetylcholine is secreted from the presynaptic neuron, and that acetylcholine is stored in vesicles, as we'll talk about in chapter 13, actually. This acetylcholine is stored in vesicles in the presynaptic membrane, in the presynaptic neuron. And these vesicles are just liposomes. Right? So on the interior of the liposome in the presynaptic neuron, there will be these liposomes, these vesicles, where on the inside we just have a high concentration of acetylcholine. And one notable feature about liposomes or structures in general that are made up of a lipid bilayer is that they can fuse together. Remember, again, the only thing that determines the radius of a liposome is how many fatty acids. So we can take liposome A and liposome B and smash them together, and they'll self-assemble into just a larger liposome. The same thing happens in cells, right? So these presynaptic acetylcholine-filled vesicles fuse with the plasma membrane of the presynaptic neuron, and when they fuse, now what was inside of the liposome becomes the outside of the cell. So the acetylcholine is released into the synapse, and the acetylcholine then binds to the acetylcholine receptors, which is just a non-specific sodium-potassium transporter, and this initiates an action potential at the postsynaptic neuron. Again, we'll talk about this in way more detail in chapter 13 and 14. But we can also use these vesicles or liposomes clinically, okay? So we can take a solution of phospholipid, right? Let's just say phosphatidylcholine or phosphatidylethanolamine, doesn't really matter, okay? And we have a solution of that at the bottom of our reaction container. And on the top, what we'll have is a solution of some molecule that we want to package inside of a liposome for reasons that we'll see in the next slide here. Well, by applying sonication, you guys ever see, those videos of jewelers cleaning the really old rings? They put it in that little container of water, and all of a sudden all that dirt comes out. That's what they're doing. It's sonication. It's ultrasonically vibrating the water in that case. But this is what we're doing here. We're ultrasonically vibrating this solution so that everything is suspended in an emulsion. Okay, when we remove the sonication, now we get self-assembly of the liposomes of the vesicles, and this traps some of the molecule that we were trying to package in these liposomes inside the liposome. In this case, say glycine, right? This is going to be packaged now inside the liposome. We can filter out the liposomes using gel filtration, for example, and now we have a solution of liposomes which are packaging something. In this case, glycine is just as an example, right? But this is how, for example, the COVID mRNA vaccine works, right? On the inside of a vesicle, what we have is our messenger RNA. Okay, now we need a way to get the messenger RNA into the cell, and there is no mechanism by which the cell will uptake messenger RNA. There is no reason why a cell would ever need to take up messenger RNA, so there's not been any evolutionary pressure in order to make a mechanism for that to happen. So if we just injected messenger RNA, will eventually be degraded by the immune system or non-enzymatically just through hydrolysis. So that would never get into the cell. Of course, we need the messenger RNA to get into the cell so that the ribosome can synthesize the antigen and present it to the immune system so it can mount a response. In order to get the messenger RNA, instead what they do is they package it inside what's called a lipid nanoparticle, which is just a liposome made up of different types of membrane lipids. Okay, here's some of them. You don't have to memorize this, of course, but again, because liposomes, in this case, the cell plasma membrane and the liposome or lipid nanoparticle are both made up of a phospholipid bilayer, they can fuse. And in this case, what was inside of the lipid nanoparticle, that vesicle, now becomes the interior of the cell. So now the messenger RNA is on the inside of the cell, can be recognized by the ribosome, and we're off to the races, right? So lipid or liposomes and vesicles can be used clinically, but they're also used biologically, naturally by the cell. Then we can measure the diffusion of certain molecules across a lipid bilayer. This is quite important, right? So here, I don't get too much into this, but essentially we have two compartments separated by a hole. On this hole, we just, using a little paintbrush, right? You literally just paint over that hole with a solution containing the phospholipids that you're testing, so say phosphatidylcholine. These will form a bilayer across that little hole, and on one compartment we have whatever we're testing the permeability to. Could be sodium, chlorine, could be glucose, tryptophan, whatever. And what you do is you simply just measure the rate at which it appears in the other container, because the only way that it had to get there was through the membrane. And what we see, right, if we look at the permeability scores, charged ions like sodium, potassium, chlorine, they basically cannot pass through the bilayer, right? They're essentially impermeable to the bilayer, which is quite important, especially in neurons. Again, we'll talk about that next week. So charged particles are impermeable to the membrane. Uncharged molecules are more permeable, okay, but For example, glucose, it is uncharged, but it still has all those polar groups in the form of the hydroxyl, so it's not especially permeable, which is also useful for the cell. And H2O, though it is obviously polar, is an exception in this case. H2O is almost completely permeable to the cell membrane, right? Reason being that the concentration of water is just so high. Okay, so this is almost free to move in between the inside and the outside of a bilayer. And this principle, by the way, is very important, especially in metabolism. Charged molecules are impermeable. Remember that. Glucose is somewhat permeable. So what's the first thing that a cell does when it imports glucose from outside the cell to inside the cell? Is it adds a phosphate group onto carbon number 6, generating glucose 6-phosphate. Now what we have is a sugar derivative that is charged. Due to that, phosphate's got a negative charge, and now it's down here with sodium, potassium, and chlorine, so it traps it inside the cell, which is what most tissue cells want to do. They want to trap the glucose inside because most tissues are very selfish. The only unselfish tissue really is the liver. The liver is the only tissue that can actually get rid of glucose because the liver's role is to maintain blood glucose levels, in part. Now, that's how the lipid bilayer looks in terms of phospholipids, but that would be useless without the membrane proteins that are also associated with those membranes. And we can integrate, or we can incorporate rather, membranes into the, or proteins into the lipid bilayer in two ways. We can have either integral membrane proteins, as is the case with A and B here, and we can have peripheral membrane proteins, okay, as is the case with E, D, and C here. And what's the difference is that integral membrane proteins are integrated into the membrane, right? It's not that they're integral to the membrane in some way, it's that they're integrated into the membrane. They span the entire bilayer, okay? The only way to separate an integral membrane protein from the bilayer is with the use of detergents like SDS, sodium reductant sulfate, sodium lauryl sulfate, which is the same thing, or soap or some other way, because this disrupts the hydrophobic interactions holding the bilayer together. So we destroy the bilayer, and if we destroy the bilayer, then there is no bilayer for the integral membrane proteins to integrate into, so they are released, right? So the only way to remove an integral membrane protein is through the use of detergents. This is different than peripheral membrane proteins, which associate with the membrane directly via non-covalent interactions such as ionic or hydrogen bonds, as is the case with C, so they interact with the polar head groups. They can also interact with integral membrane proteins, as is the case with D here, or they can be covalently modified to mimic a fatty acid, or a membrane lipid rather, they can be covalently modified to mimic a lipid, membrane lipid, right? In this case we call it lipid anchor. And if we look at the populations of proteins making up or present in the cell membranes of different tissue types, we see that there are many different configurations. So for example, what we have here, A is an erythrocyte, B is the rod cell of the retina, and C is in muscle cells. So this is present in the sarcoplasmic reticulum membrane. And what we see, if we do a Western blot, or rather an SDS page blot, and stain it with Kumasi, what we see is, so we isolate the membranes from these three different tissue types, and we do a SDS page, as we talked about in chapter something, okay? So we can see here the identity, or the number of proteins that are present in this case, the red blood cell. So we see the red blood cell membrane has many proteins on the surface. The rod cell in the eye only really has one, okay? Because again, what's the purpose of the rod cell? All the rod cell has to do is to react to photons and to transmit that to the visual center of the brain, right? So this really only needs one thing present on its surface. Of course, it needs a sodium pump and potassium pump and a couple other things, so we see that over here. So these have two vastly different functions, right? The red blood cell and the rod cell, and we can see that the membranes that we find, or the proteins that we find in their membranes, are equally different. Okay, so depending on what that cell is doing, it will express different proteins on its cell membrane in order to accomplish that purpose. Here, the membrane of the sarcoplasmic reticulum is going to have a high concentration of, for example, the Circa pump, which pumps calcium into the endoplasmic reticulum lumen, right? So this is not going to be present on any one of these, right? Because these don't have a reason to do that, and so on and so on. So So we look at integral membrane proteins, right? One of the ways that proteins can integrate, oh yeah, so
What did you say was covalently modified?
a protein that integrates, a peripheral membrane protein can be covalently modified to mimic a membrane lipid. And we'll see that in just a second, right? So this is an example of a peripheral membrane protein because it does not span the membrane, it's only present on one side of the membrane. So it counts as a peripheral membrane protein. Now, one way that integral membrane proteins can span the membrane, a very common way, is they're used through a 7-TM domain, or 7 transmembrane domain. This is made up of 7 alpha helices, each of which at least partially spans the bilayer. And because they have to make contact with the nonpolar tails of the membrane lipids, the residues making up these alpha helices are going to be mainly hydrophobic, right? They're going to be mainly nonpolar. Remember from chapter 2 how an alpha helix looks in 3 dimensions. Remember, the side chains of the residues making up that alpha helix always point out away from the axis of the helix, okay? So if we have many nonpolar residues making up these helices, well, now we have many nonpolar functional groups making contacts with the nonpolar tails, so that's allowed, that's good. So if we look here at the sequence for Bacteroideopsin, which is a 7-TM receptor in this case, so it's a receptor made up of a 7-TM domain, what we see is we can look at this sequence here, which corresponds to the first alpha helix in our 7-TM domain, tryptophan, isoleucine, tryptophan, leucine, alanine, right? All these hydrophobic residues. Here we have polar residues somewhat, right? Actually, no. Well, serine, right? Here we have an aspartate, a lysine, right? So these will make up these loops down here that still have to make contact with the aqueous environment, the cytosol or the intracellular environment, followed by another stretch of nonpolar residues making up the second helix. Here we have some more polar residues helping to, make those loops. Here another stretch of nonpolar, and so on and so on. And so these stretches of nonpolar alpha helices can integrate into the membrane and anchor it there, because now the energy required to move that protein, right, these 7 hydrophobic transmembrane helices, the energy required to move that from inside the membrane to an aqueous environment, so either inside or outside of the cell, is large, right? Because now you're taking away favorable interactions between the nonpolar helices and the nonpolar tails, and you're replacing them with unfavorable interaction between the nonpolar helices and the polar environment, right? So these are now stuck permanently inside the double bilayer here. We can also integrate into the membrane using beta sheets, right? Beta sheets are another way that we can integrate. Here what we're looking at is an enzyme or a protein called porin, and it forms essentially that. It's a pore in the membrane, okay, that allows charged molecules to flow through. And how does this work? Well, recall again the structure of a beta sheet, right? We have an alternating pattern of up and down, up and down, up and down. So if we look at one beta sheet, right? Pretend my arm here is a beta sheet. Here, first residue points up, second residue points down, third residue up, fourth residue down. So we have this alternating pattern so that one side can be mostly polar and the other side can be mostly non-polar. And that's what we see happening here. One side of each of these beta strands making up this barrel structure, right? These beta strands are all hydrogen bonded together, of course, because the hydrogen bonding occurs between the backbone nitrogens and carbonyls, right? But this side of this beta strand will be nonpolar. The opposite side will be polar. So if we look at it from the top, this side here will be nonpolar, and this side over here will be polar. And the same for this beta strand and this beta strand and this beta strand. The effect being that on the outside of this barrel, we have mostly nonpolar residues which can interact with the nonpolar tails, and on the inside we have mostly polar residues which can interact with polar molecules like, say, potassium or sodium, right? So these form pores in the nonpolar membrane that allow polar molecules to flow through, and we'll talk about that when we get to channels. And if we look at the sequence of each of these beta strands, so here's beta strand 1, beta strand 2, beta strand 3, what we see is this alternating pattern of polar, nonpolar, polar, nonpolar, polar, nonpolar, so that the inside is mostly polar and the outside is mostly nonpolar, so it can integrate into the membrane here. We can also get interacting or integral membrane proteins that don't necessarily span the bilayer, right? These are kind of an in-between between the two. So for example, prostaglandin H2 synthase, which generates prostaglandin H2, which is associated with inflammation, okay, is an integral membrane protein, but it's integrated via only a portion of its amino acid structure, right? So here we have these nonpolar helices, which interact favorably with the nonpolar tails in the bilayer, but the rest of it is free to be an aqueous solution, okay? So we have this aqueous protein that is kind of anchored in a way to the bilayer, okay? And what happens here is prostaglandin H2 synthase takes the fatty acid arachidonase, right, this specific fatty acid, and it converts it via cyclooxygenase activity and peroxidase activity into first prostaglandin G2 and then prostaglandin H2. Actually here, at this cyclooxygenase activity is where aspirin, ibuprofen, all different kinds of non-steroidal anti-inflammatory drugs, NSAIDs, act. Okay, they inhibit the cyclooxygenase activity of prostaglandin H2 synthase, and they prevent the formation of prostaglandin G2, which of course then can't lead to prostaglandin H2, thus reducing inflammation. This is how these NSAIDs work. In fact, you might hear these NSAIDs, specifically a subset of NSAIDs, be referred to as COX-2 inhibitors, right? That's what the COX is referring to, cyclooxygenase, COX-2. Right? These COX-2 inhibitors prevent the formation of prostaglandin G2 and thus lead to reduced inflammation. Yeah, I guess we'll talk about this. And the way that these COX-2 inhibitors work, for example, acetylsalicylic acid, aspirin, okay, they work by forming something that we would call a suicide inhibitor, okay? The way that a suicide inhibitor works is by covalently modifying either a critical residue in the active site or somewhere else in the enzyme in order to prevent that enzyme from catalyzing any further reaction. So in this case, the prostaglandin H2 synthase is a critical serine at position 530 in the active site of prostaglandin H2 synthase. This can react with acetylsalicylic acid and transfer the acetyl group from aspirin onto the terminal hydroxyl of serine to form this acetylated serine derivative. And because that serine is critical in forming prostaglandin G2, right? By removing it essentially, we now eliminate this enzyme's ability to form prostaglandin G2 and thus reduce inflammation. Now, peripheral membrane proteins, again, they can interact with the bilayer either through non-specific or non-covalent means like prostaglandin H2 synthase, or they can be covalently modified to behave as if they were a membrane lipid. And these come in three major categories. We can have a palmitoylcysteine modification, we can have a farnesylcysteine modification, or we can have a glycosylation derivative, right? So let's talk about the two simplest ones first. So S-palmitoylcysteine, recall that cysteine ends in a sulfhydryl. So what we can form is a bioester with the terminal sulfhydryl of a cysteine molecule in or on the surface of a peripheral membrane protein, right? So here we have our protein, here we have a cysteine on the surface, and we can add a fatty acid forming a thioester here, and this can now just integrate directly into the plasma membrane. So it behaves as if it were a membrane lipid. And the same is true for this farnesyl modification. This is a C-terminal S-farnesylcysteine. In this case, specifically, we modify the terminal sulfhydryl of a cysteine at the C-terminus of the protein, right, with this farnesyl group here. This farnesyl group, very much like S-palmitoylcysteine, just makes it behave as if it were a lipid and integrates into the membrane. More complicated in structure, but more interesting, is the GPI anchor, the glycosyl phosphatidylinositol. right? So right off the bat, we know that we have phosphatidylinositol, which is already a membrane lipid. It's a derivative of phosphatidate, where the alcohol is simply inositol, right here, right? So here we have our glycerol, our two fatty acids, phosphoryl group, and inositol. One of the hydroxyls on inositol is, of course, in this phosphodiester bond with the glycerol, but one of the other hydroxyls, recall that that inositol has like 5 hydroxyl groups, okay, is in a glycosidic bond with a sugar residue, which is in a glycosidic bond with other sugar residues, which is then attached to the C terminus of a protein. So in this case, not only is it acting as if it were a membrane lipid, it is literally a membrane lipid where the polar head group happens to be a protein or enzyme, right? And it integrates into the membrane. Now we can predict transmembrane helices specifically using the hydropathy index. The way we can do this is by understanding that, yes, of course, the residues that make up a transmembrane helix must be hydrophobic in order to interact with the nonpolar tails of the membrane lipids. But we also know that there has to be a stretch of nonpolar residues. So it can't just be, overall is the protein polar or nonpolar. We have to have a stretch of residues that is going to be mainly nonpolar, so that the entire helix is nonpolar, so that it can integrate into the bilayer. The question becomes, how big does that stretch have to be? Well, it turns out that the phospholipid bilayer is about 30 angstroms wide. And remember, the pitch or rise of an alpha helix is 1.5 angstroms per residue, right? So each residue rises 1.5 angstroms. So in order to span the full 30 angstroms of the phospholipid bilayer, we need 20 residues. 20 times 1.5 gives us 30 angstroms. So let's look at windows of 20 amino acids, and let's calculate how hydrophobic they are. Now we can do this intuitively just by looking at, is it an alanine, is it a leucine, is it a phenylalanine, and so on, right? But there's a more quantitative way to do this by using what we call the transfer of free energy. And this is essentially the amount of energy that's required to move a molecule or a mole of molecules from a nonpolar environment into a polar environment. So in the case of phenylalanine, right, it will require 15.5 kilojoules to move 1 mole of phenylalanine from a nonpolar environment into a polar environment, which makes sense because we're going from a nonpolar phenylalanine in a polar environment and moving that into the less favorable configuration of having nonpolar phenylalanine in a polar environment. Okay, so that's going to require the input of energy. Conversely, aspartate, right, that's going to release 38.6 kilojoules for every mole if we move it from nonpolar into polar, because aspartate, of course, is very polar. It's got 2 carboxyls in the alpha amino group. It's extremely polar. So the more positive number, the more positive transfer free energy, we can categorize that or estimate that to be how hydrophobic a residue is, right? So here we have the transfer free energy for all 20 residues. So now if we have the sequence of our protein, okay, and we look at, all right, let's count up and add all of the transfer free energy starting from amino acid 1 to amino acid 20. What does that come out to? All right, that's what we're going to count at that position, right? That's our first window. Then we slide that window 1 to the left, or 2 to the right? So now we're looking from amino acids 2 through 21, and then from 3 to 22, and so on and so on and so on. We keep shifting this window and adding up all of the transfer free energies to come up with this hydropathy index plot that we see here, right? The position here is the first amino acid residue in the window, right? So we're starting at position 1 through 21. We add up all those. That's what we're going to plot here, and so on and so on and so on. Empirically, what we observe is that once we reach a criterion level or a threshold of about 84 kilojoules per mole, right? If a 20 residue long window has that hydropathy index, that's likely to be a transmembrane helix. So in the case here of blanking on the name, whatever, it doesn't matter. Do I have it on the slide somewhere? Glycophorin, thank you. So here, what we have is the amino acid sequence for glycophorin, right, starting from the N to C, and this is the hydropathy index plot for glycophorin. And what we see is that we get this peak above the threshold level, starting at position about 70 to 75, somewhere in there, okay? So, starting at position 70 to 75 somewhere, there's likely to be some kind of transmembrane helix. Right? So that's the prediction. But if we solve the structure of glycophorin in the membrane, what we can see is that indeed at position 7D, 1, 2, 3, right, we begin this transmembrane helix. Right? So predicting transmembrane helices is simple using this hydrophobe helix. Now we can't do the same for example, porin. We go back a couple of slides here. Porin, we can't do the same thing because in this case we specifically don't want a stretch of nonpolar residues, because that would indicate that both sides of each of these beta strands will be nonpolar. We don't want that. We want one side to be polar and one side to be nonpolar. So in this case, we specifically want an alternating pattern. So in this case, the hydropathy plot would be closer to 0, right? Instead of all the way up here at 84 kilojoules per mole. So this method here for predicting transmembrane components only applies to alpha helices. You can't apply the same logic, obviously, to a beta barrel or anything like that. Now, we know a little bit about the membranes, and we know about the proteins that make up these membranes. So how do they function together? How does it look? Well, this makes up what we call the fluid mosaic model. Right? Does anybody know what a mosaic is? Right? Those kind of fancy tile works that make up pictures and all this, right? So it's tiles on mortar, and different colors and shapes are put together to make an overall picture. And that's the same thing that occurs in the plasma membrane, right? In these lipid bilayers, the cell membrane. We have the mortar, which in this case is the membrane itself, and we have the tiles, which in this case are the proteins. But it's a fluid mosaic because these proteins are not fixed in place as they are in a regular mosaic by the mortar. Instead, these tiles or these proteins are free to diffuse laterally all throughout, okay, all throughout the membrane. Mostly, there are some cases where they're fenced in or anchored to the cytoskeleton, but overall, right, they're free to diffuse laterally within the plane of the membrane, right? This is called lateral diffusion, and this is why we call it a fluid mosaic. It's a mosaic that's free to move around, okay? Transverse diffusion is rare, okay, where we have a protein, or let's just say a fatty membrane lipid, right, on one leaflet that flips over to the other leaflet. This is rare, again, because now the polar head group has to pass through all of this non-polar space, so that's very rare. But this lateral diffusion of either proteins or the fatty acids making up that particular leaflet, lateral diffusion is essentially free, right? We're not breaking any, we're not net making or breaking any interactions, right? We're breaking non-covalent interactions with some fatty acids, but we're forming them when we move something else. So this is free, this happens freely. What we never, ever see, has never been observed, as long as this has been studied, is the transverse diffusion of a protein, right? So fatty or lipids, the membrane lipids can diffuse transversely, rarely, but a protein will never transverse, okay? It will never do that. And this is good for the cell because, again, remember, membranes are asymmetric. Okay, so we want to make sure that what's on one side of the membrane stays there. Okay, that helps to differentiate one side from the other, which is important because that helps to set up the polarity of the membrane, right? Which direction different molecules should move and so forth, right? So transverse diffusion never occurs with proteins. And how is this proven? Actually, very interesting experiments here called FRAP, fluorescence recovery after photobleaching. And what this essentially does is it attaches a fluorophore, which is a small molecule that emits light, right? So say that we want to measure how free to diffuse through the membrane protein X is, right? Well, we can synthesize protein X onto which we covalently attach this fluorophore. So now protein X should always be glowing green, let's say, okay? Now these fluorophores can be destroyed. They can be destroyed by high energy light, like from a laser. And this process is called bleaching. So now we have all these protein X's that are carrying this fluorophore on the cell surface, okay? And they're not going to go the other way. They're not going to diffuse transversely, so they're free to diffuse all throughout, okay? After we bleach a small area of the plasma membrane, well, that destroys the fluorophore in this region. And what we observe is that the fluorescence in this region actually returns. okay, it recovers. So this is fluorescence recovery after photobleaching. Now we've destroyed the fluorophore. There is no coming back from that. So the only way that fluorescence can be recovered in that area is if the photobleached molecules moved out of that area and the unphotobleached molecules moved into that area. So we've got diffusion out and diffusion in, right? The time that it takes to do that determines how free to diffuse that protein is, right? And we can say that the average distance moved, S, by a protein in the plane of the membrane is equal to the square root of 4 dt, where t is time, d is the diffusion coefficient, and s, again, is the average area traversed, okay? So this equation is also in your book. The higher the diffusion coefficient, the higher the d value, right, the more free to diffuse that protein is, right? So you might imagine a very small protein will have a high d value because it's got less mass to move around, so it's free to diffuse more than, say, a large protein. A large protein has more mass, so it moves more slowly, so you will have a smaller d value, smaller diffusion coefficient. Fatty acid composition determines also membrane fluidity. So of course, the membrane lipids are made up of the 2 fatty acids, right? In the case of phospholipids, or the 1 fatty acid, in the case of sphingolipids and glycolipids. Okay, but the composition of these fatty acids, or the identity of the fatty acids making up the membrane lipids, determines that membrane fluidity. In order to do this, first let's discuss the TM, or the melting point of different fatty acids, right? And what effect the structure of those fatty acids has on the TM. So TM is basically the point at which, the temperature rather, at which that solution, or that, not solution, but that, say, reaction container of a specific fatty acid is halfway in between being an oil and a fat, right? So here we have fluidity on the y-axis, so on the bottom we have less fluidity, so we're more solid-like, so we're behaving more like a wax or a fat. At the top, we're more fluid-like, so we're behaving more like an oil. Okay, so obviously as you decrease the temperature, now you'll start to freeze it in a way, right? So you'll start to see it behave more like a wax. As you increase the temperature, well, now it behaves more fluidly, right? The point at which we're at 50% solid, 50% fluid, that's the TM. In other words, above the TM, we're an oil, and below the TM, we're a solid, or a wax fat type of thing, okay? And let's look down here at the different TMs of different fatty acids. So we have an 18 carbon fatty acid saturated. That's got a TM of 58. That means that above 58 degrees, we're a fluid, and below 58, we're a solid. Right, so let's say that we have a bottle full of stearate, and you put that in the fridge, it's about 4 degrees Celsius. Okay, well now that stearate is going to be a fat. It's going to be waxy solid-like. But let's look down here at oleate, which is 18 carbons still, right? So 18 carbons of stearate, 18 carbons of oleate. The difference here being that we're a cis delta 9, so we have 1 unsaturation. Okay. Well, now the TM is minus 22. Right? So we put that in the fridge, which again is 4 degrees Celsius. That's going to stay an oil, right? Because 4 degrees is higher than negative 22. Right? So what do we see? Well, the degree of unsaturation has an outsized role on the melting temperature of a fatty acid. Same number of carbons, right? The only difference here being that one has a double bond. We'll see why that is in just a second. But another thing that can affect the melting temperature is the number of carbons, but to a much lesser extent. So if we look at 2 saturated fatty acids, stearate and palmitate, okay, the only difference here is 2 carbons. Well, stearate has a TM of 58, while palmitate has a TM of 41, okay? So stearate behaves a little bit more like oleate in a way, right? So it's more fluid-like at the same temperature. This has to do with the number of carbons because in a longer chain, there are more carbons to make van der Waals interactions, okay? Fewer carbons, you have fewer van der Waals interactions, so they don't hold together as strongly. So if we look at here, right, here would be an example of, let's say, stearate or palmitate, okay? We have a saturated fatty acid, it's fully linear, So these can stack on top of each other very nicely and form very nice van der Waals interactions between them. And so we get these pseudo-crystalline-like structures, which result in a solid-like appearance, okay? But once we start to introduce cis double bonds, right? Well, now we have much less contact between them, right? Here, this whole region of this unsaturated fatty acid is not making any van der Waals contacts, okay? So these don't have as many non-covalent interactions holding them together, and so they're more free to slide apart from each other. The kinks in the cis unsaturations, right, they prevent this crystalline-like structure from forming, and so it behaves more like an oil. Here what we have is a trans unsaturated fatty acid. A trans unsaturated fatty acid behaves very much the same as a saturated fatty acid because they have the same structure. So in fact, if we look at, it's not listed here, but if we look at palmitate, right, versus a palmitate with one unsaturated double bond, right, in the trans configuration, the Tn would be almost exactly the same. It would be something like 38 degrees, okay, because we are missing some interactions, but not much, okay? So trans unsaturated double bonds behave almost exactly the same way as their saturated counterparts. But we don't encounter trans double bonds very often, at least naturally, right? Where do we encounter trans double bonds is in foods, right? Not anymore, actually, but the reason was that saturated fatty acids actually are more shelf stable. So back in the 50s, some intrepid industrial scientists figured this out. If you saturate a fatty acid, it's more shelf stable, right? Especially in the 40s and 50s, where shelf stability was very important, right? They said, well, fuck it, let's just saturate all the fatty acids. Okay, and the way that they did this was chemically was just by filtering hydrogen gas over a palladium catalyst. Was it platinum catalyst, right? And this saturates all the double bonds. So you end up with saturated fatty acids. How margarine was first made, by the way. As it turns out, an unexpected side effect of that process was not the saturation of the fatty acid, but instead an inversion from a cis double bond to a trans double bond. So in addition to saturated fatty acids, you had a lot of these trans fatty acids, which are not necessarily inherently bad, right? They're functionally the same as a saturated fatty acid, which aren't, great for you, but they're not dangerous, all right? So what is the deal with these trans fatty acids? Well, as it turns out, for reasons that are still not entirely well understood, trans fatty acids in the body interact with the liver in such a way that it increases the production of LDL cholesterol, low density lipoprotein, right? That's the bad cholesterol. It can lead to plaque formation in the blood vessels, which constricts them. It's going to lead to edemas or cardiovascular disease. So it's not anything inherently wrong with the structure of a trans fatty acid. It's just that they react in an unexpected way to the body in order to increase the amount of an undesirable side product, in this case LDL cholesterol. Right, so you can see trans fatty acids, there used to be a lot in margarine, right? Of course, it's how margarine was made through saturated fatty acids, but now, well, not now, 20 years ago, they changed the way that they make margarine in order to not produce any of these trans fatty acids. The same for vegetable shortening, like Crisco, right? So we used to fry chicken in Crisco, and it was super good, and they got rid of the trans fatty acids. Now it's not quite as good, right? That's right. Should be eating Crisco fried chicken anyway. All right, now we're gonna finish off just talking about cholesterol, and then we'll call it a day. So cholesterol, again, we kind of is the bastard child in a way of membrane lipids. Not because they're not important, but because they're not what we think of when we think about membrane lipids as, the circle with two tails. They're vastly different. And what cholesterol does is to help to regulate membrane fluidity. And it does so in kind of a weird way. It's almost counterintuitive in a way. We look at membrane fluidity, right? Again, going from solid-like at the bottom to fluid-like at the top. Here, without cholesterol, the blue line, we see that we get a very quick transition from mostly solid-like at low temperatures to mostly fluid-like. And where we want to be is somewhere in here. So our membrane lipids can tolerate very little temperature difference, right? Going from up and down before they become either too solid or too fluid, right? So this slope here is quite steep. With cholesterol, this line becomes more or less linear, which means that at low temperature, we are more solid with or more liquid rather with cholesterol, which is good, right? And over here, okay, at high temperature, we're more solid with cholesterol, right? So it goes from having an effective temperature range from about here to about here to being able to go from probably around here to over here. So it widens the allowable temperatures, right? How does it do this? Well, at low temperatures, okay, where we're more solid, like cholesterol inserts itself into the membrane, okay, it acts as if it were a membrane lipid, but it doesn't provide very good van der Waals contact, okay, because of that hydrocarbon at the end there. Okay, it doesn't provide very good van der Waals contact, and so it disrupts those van der Waals contacts between the fatty acid tails, right? So they're free to move in and out, right, or back and forth. Right, so they behave almost as if they were cis unsaturated fatty acids. At high temperatures, though, they do the opposite. They provide more van der Waals contact than they would than the fatty acids would normally encounter. So it holds them together. So it either disrupts van der Waals at low temperatures, or it provides van der Waals at high temperatures. And in doing this, right, now it can help to make membranes more fluid at low temperatures and less fluid at high temperatures. So we achieve optimal membrane fluidity over a wider range of temperatures or fatty acid conditions. All right. So do we need to do this? No. All right. So slides 36, 37, 38, 39. So starting from here on, we've already talked about basically. So we don't have to really get into that. Here's just showing where. We'll talk about this later in the semester. All right, good. So we'll leave it there.